We will discuss about acidic strength among phenol, ortho, meta and para cresol. So first see the structure of phenol, orthocresol, metacresol and paracresol. When methyl group is attached at ortho position with phenol, then it's called orthocresol. When at meta, then metacresol, and when at para, it called paracresol. Since we know methyl group, that is alkyl group, is electron releasing group or electron donating group. So this group influence plus I effect in the benzene ring. Plus I effect always decreases. Plus I effect decreases acidic strength. Why? Because plus I effect influence electron density in the chain, due to which OH bond becomes stronger, and due to strong OH bond, release of H plus decreases. Due to decrease in the intensity of H plus released, acidic strength ultimately decreases. What is acidity or acidic strength? Since we know when a compound releases protons in its solution, it's called an acid. As per Bronsted-Lowry concept, when a compound releases proton in its solution, it behaves as an acid, means acids are proton donor. That's why this Bronsted-Lowry concept is also called as protonic concept of acid and bases. So as per Bronsted-Lowry, those that releases proton H plus in their solution behaves as acid, since phenol has tendency to release H plus and it forms phenoxide ion. That's why it behaves as an acid, C6H5O negative plus H plus. Phenoxide ion is formed due to release of proton in its solution. Phenol behaves as an acid. Since between phenol and rest of the ortho, meta and paracresol, phenol has no any group attached with the chain with the benzene ring, while here in ortho, meta and paracresol, methyl group is attached. Since methyl group behaves as electron releasing group that influence plus I effect in the chain, due to which acidic strength decreases. So it is clear that ortho, meta and paracresol are less acidic than phenol. So first thing clear from this explanation, phenol is more acidic than ortho, meta and paracresol. It is due to presence of methyl group in cresol. It influences plus I effect that decreases acidic strength of the ring. That's why phenol is more acidic than ortho, meta and para cresol. Next thing, it was the explanation for the first half of the question, that is phenol, ortho, meta and paracresol. So phenol is more acidic than rest of the cresols, ortho, meta and para. But what will be the acidic strength among ortho, meta and paracresol? So for this we will discuss here about the resonating structure of phenol. when it releases proton in its solution and forms phenoxide, and what will be the resonating structures? So let's see here. This is phenol. In alternate double bond in benzene ring is present with OH. Oxygen has two lone pair of electrons. When it releases proton, takes the electron and H plus moves out. So formation of phenoxide ion takes place like this. It becomes single bond O with negative charge because the covalent electrons are taken by more electronegative oxygen. Here, delocalization of these lone pairs takes place between oxygen and carbon and these pi electrons to the ortho position. So, a resonating structure formed will be like this double bond O, and it becomes with lone pairs and it becomes with negative charge, and the rest structure will be as such. But due to influence of this delocalization of electrons, again these negative charge converts into pi electrons and pi again into the lone pair of electrons. So it becomes double bond O with lone pair of electrons at oxygen. Here it converts into pi and it becomes negative at para position, and this pi electron as such. Again, next structure resonating is formed. That next resonating structure will be like this: benzene ring with double bond O with lone pair pi bond. It becomes pi bond and it becomes lone pair at ortho position again. So pi, and it becomes lone pair with the negative charge at ortho position. Overall, we can say that in whole benzene ring, partial double bonded structure along with partial double bonded carbon oxygen with partial negative charge at ortho and para position is observed. I repeat this resonance. Due to resonance, the lone pair and pi electrons delocalizes. That's why this resonance is called lone pair pi interaction. Either it will be lone pair or lone pair pi interaction. Here in phenoxide, and the resonant type is lone pair pi interaction is carried out, due to which different resonating structures are found that ultimately results resonance hybrid. And this resonance hybrid has partial negative charge at ortho para position with the oxygen that is attached with the proton. Now, since it is clear that higher electron density is present at ortho and para position, that's why incoming methyl group, that is electron releasing group, will accommodate favorably at meta position. Meta position will be much better for attachment of methyl group due to less electron density as compared to the ortho and para position. This methyl group, when attached with meta position, it will influence a very little plus I effect, due to which a little increase in electron density takes place at oxygen that attached with the protonic hydrogen H plus. I repeat again, higher electron density is present at ortho and para position. Since methyl group has tendency to release electrons to influence electron density in the ring due to plus I effect, Since meta position is much favorable for attachment of methyl group, so at this position, after attachment of methyl group, there will be very little increase in electron density at this oxygen. That's why this meta cresol will be less acidic than phenol but more acidic than ortho and para. So next order we can note down phenol is more acidic than metacresol. But what will be the relation between ortho and paracresol? We will discuss later on in this segment. Here, at meta position, attachment of methyl is much favorable because higher electron density is present at ortho and para position. Due to attachment of this methyl at meta position, there will be a little increase in the electron density at oxygen and it will be less acidic than phenol but more acidic than ortho and paracresol. But when we will see the structure of ortho and para, let's see ortho. This is orthocresol and this is paracresol. Since from this resonance hybrid, it's clear that when methyl will attach at ortho and para position, higher electron density will be at the oxygen attached with the hydrogen. Since we know plus I effect also depends on distance, closer the group attached, higher will be its influence. Its influence will be higher. So the comparative analysis between orthocresol and paracresol depends on the distance of the methyl group attached. Since in ortho methyl group is attached close to the OH, it provides higher electron density at oxygen that results more stronger OH bond as compared to para. Para is just opposite to the oxygen attached and it provides less electron density at oxygen as compared to ortho. So higher the electron density due to closer group attached, stronger will be the OH bond and less tendency will be to release proton in its solution. That's why orthocresol will become less acidic than paracresol. I repeat, due to higher plus I effect in orthocresol because methyl group is attached very close to the oxygen and increases higher electron density at oxygen due to which OH bond becomes stronger. Tendency to release H plus decreases that results decrease in acidic strength. While in para it is attached, methyl group is attached opposite to the oxygen, so less electron density is present at oxygen in para as compared to ortho, due to which this OH bond will a little weaker as compared to the orthocresol and it releases proton more quickly as compared to orthocresol. So acidic strength of paracresol will be higher than orthocresol. That in first half we have noted down that phenol is more acidic than metacresol. It was the first half answer, and in the second half we know paracresol is more acidic than ortho, therefore meta will be more acidic than paracresol, and para will be more acidic than orthocresol. This will be the decreasing order of acidic strength. Decreasing acidic strength from phenol to orthocresol. Phenol will be more acidic than rest of the cresol, whether they are meta, para, or ortho. In para and ortho, due to distance, due to distance, higher influence of plus I is observed at ortho. That's why it becomes less acidic as compared to para. So least acidic among these will be orthocresol and highest acidic will be phenol. So this is all about the acidic strength among phenol, ortho, meta and para cresol. Thank you.